Hmm. Hello. 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 Ayush, am I audible? Yeah. Okay. 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 So now, yeah, others are also joining. So, but how are you all? I mean, are you all fine? Hello. Are you all fine, beta? Yeah. Okay. Okay. But your voice is too low, Prachi. I don't know why, but maybe some technical issues. Okay, so this is Atul Kumar, and I'm your tutor for the day. And today we'll be dealing about the fundamental unit of life, and this is session 3. Okay, so we'll be dealing about the session third of this chapter. Now, introducing you to this platform, you'll be having 4 or 5 options. First, you'll be having this audio and video option. So, if possible, better please keep your audio muted, and yeah, try to turn on your videos as well. Okay. Yeah, very good. Thank you. Thank you. Others also, if possible, please turn on your videos so that we can interact while seeing each other's faces, each other's expression, and that is really excellent for us. Then we'll be having that chat box. So in that chat box, you can always ask your doubt or anything of that kind. Maybe some answering my questions or anything when you that you want to interact with me. Then you'll be having that hand raise option. So in case you feel like we need to ask something in your audio only, you can always unmute and ask yourself while raising your hand. Whenever you will raise your hand, I'll be taking up a comfortable pause and then take up your questions or query. Okay. Yeah, good evening, beta. Then we also have this.
Yes, sir.
Yeah, good evening. Then one more thing is that troubleshoot option. So in case you feel like any technical issues is there at your end, you can always go to that troubleshoot option and get it sorted. Now, talking at your attendances, I can see it's Lakshya and then Sahana. They have their 100% attendance, and others, all others, maybe 1 or 2 days. Uh, yeah, there is one who has got several days of absence. So, beta, please, 1 or 2 days is then too okay, but I mean, several days in just 10 days is not acceptable. Please try to be more consistent. Okay. And yeah, I guess this is our second or third session. No? We are meeting for the second or third time, isn't it?
Third session.
Third. Now, talking about your attendances, I mean, as I said, please be regular, more attentive, and more consistent. Now, I can speak both English as well as Hindi. So, if you feel like you need to ask something in Hindi also, I mean, I can see your primary language is English, but still, if you feel like that, okay, this topic or this much thing can be explained in Hindi as well, and it would be much more better for me, then you can ask. I mean, at your concern that okay, sir, please explain it in Hindi also. I'll be explaining it in Hindi also for you only, and others I'll be explaining it in English also. Now let's begin with the session. And before beginning, let me first revise what we all studied. We talked about that cell wall. We know, I mean, in our previous sessions, from our previous sessions, that cell wall is the protective layer in plant cell. it provides mechanical strength and support, and it is freely permeable. Okay, then we talked that okay, it's not like only plant which is made up of cell. There are many other organisms or many other groups of organisms also which have cell, but in plants the cell is made up of cellulose. In other groups like fungus, cell wall is made up of chitin. In algae, the cell wall is made up of cellulose, while in bacteria, the cell wall is made up of peptidoglycan. Not talking about that cell membrane. We know that cell membrane was a phospholipid bilayer made up of phosphoric acid and lipid bilayer, as there are two layers of it. Two layers, that is why called bilayer. There is a transport protein, the gate-like structure that allows the molecules to move through it. Okay. Then we talked about the types of transport on the basis of energy demand. If energy is required, it is called active transport, and if energy is not required, it is called passive transport. Now, active transport is of two types: endocytosis and exocytosis, while passive transport is again of two types: diffusion and osmosis. Okay. Then, talking about this types of solution. Yeah, Lakshya said, what's your doubt, beta?
Can you explain exocytes again?
Okay, so actually, in that slide in our that session, we talked about active, in which we talked about active transport. We first discussed a term called phagocytosis. Can you memorize it? What is phagocytosis? Anyone? What is this phagocytosis?
Sir, it is a term related to amoeba.
Okay, phagocytosis actually means engulfing of anything. Engulfing of substances is called phagocytosis. Okay, so if you are engulfing something inside your cell, you call it as endocytosis. And when you engulf something out, I mean, just like vomiting, then you call it as exocytosis. Okay, so engulfing something outside your cell is called exocytosis, which is analogous to that vomiting. Got it, beta? Puneet? Was it?
Yes, sir.
Okay, sorry, it was Laksh. Okay, sorry. Yeah, done. So now let's discuss about the types of solution, different types of solutions. So there are three types of solutions: hypertonic, hypotonic, and isotonic. All these three names are in accordance to the amount of solute, the solute concentration inside the cell. Hypertonic, the solute concentration is higher. Hypotonic, the solute concentration is lower. While in isotonic, the solute concentration is equal to that of cell. In hypertonic solution, the size of cell will decrease, it will shrink. In a hypo, the cell will become hippo, it will swell up. While in isotonic solution, there is no change in shape of cells. Okay, now we are to a poll question, and let me see who among you is answering me the first. Okay, I'll be giving you 25 seconds for this. Just give it a try, beta. Your time starts now. Why is cell membrane called a selectively permeable membrane? 20 more seconds left. Yeah, it's Pius. Very good, Pius. Then Laksh. What about others? Then Shreya, then Sahana. Others, give it a try, beta. Only 4 of you are still left. 5 more seconds, then I'll be moving to the answer slide. Others, give it a try. Everyone. Okay. No one other? No one else? Okay, so let me move to the answer slide. The correct answer to this question is B. I mean, Pius must be regretting. Beta Pius, I mean, why are you confused? I mean, what is the doubt that that compelled you to change your answer? What were you thinking? Pius, ask it so that I can clarify it. Okay, for Indrani, the screen was white. Okay, beta. Try rewinding using Chrome or some other, browser, and this join while using learn.byjus.com, and then turn on desktop mode in that browser. Yeah, Pius, what was your doubt, beta? Why did you change your answer? I mean, anything that you need to clarify? Okay, so he left. No issues. Maybe there was there would be technical issue, or he might be joining again. Maybe. Let's see. So the correct answer is that, okay, if cell membrane is allowing only selected molecules to pass through it, these types of cell membranes are called selectively permeable membrane. If the membrane is only allowing selected molecules or selected particles to cross through it, it is called selectively permeable membrane. Okay. Now again, there is one more question. This time the question is rate of diffusion across cell membrane depends upon. Okay, so it depends upon two things. First, it depends upon the size of molecules, and it also depends upon,
Sir, both A and B.
yeah,
C
yeah,
option.
yeah, very good, Laksh. And it also depends upon solubility of in lipids. Okay, two things. Size of molecules, the more smaller is the substance, the more easy for it to cross that membrane. And solubility, if it is of same nature as that of membrane, then it can again cross easily. For example, if the membrane is made up of water, so is it possible for something like oil to cross through it? The answer is no. It would be very difficult for an oil to cross a water. The same is also true with vice versa. Okay, so lipid is also something which is similar like that fat. Okay, not exactly same, there are differences. So lipid is the main constituents of cell membrane. So anything like water would not cannot easily cross this membrane. So it also depends upon the solubility rate of diffusion, and so the answer is C. Now let's again go back and visualize a cell. How to visualize a cell. In last class, we discussed about how to. What is a microscope? The structure, the labeling part of that microscope. In today's session, we'll be working on how to use that microscope, how to visualize a cell in that microscope. Okay, and for that, I'm calling our master tutor to explain it better.
Experiment together and see cells under a microscope. So we're going to do this experiment on an onion, okay, because I can't really cut up anything else. So here are the things you're going to need. First, take a watch glass and pour some distilled water into it. Take a peeled onion, cut it up into half. Now what I want you to do is separate one layer of the onion. Use forceps to separate the really thin epidermis from the inner part of the onion layer, and this is what you're going to actually observe. This transparent layer is very, very delicate and needs to be handled very carefully. Transfer this epidermis into the watch glass with the distilled water. Swirl it around gently to clean it. Now, take the saffron and stain. Add a few drops into a second watch glass which doesn't have the peel. Take a paintbrush. Transfer the peel into the watch glass containing the stain. Leave it there for 30 seconds. Allow it to, take in all the stain. Then transfer it back into the water. Next, pick up a glass slide. Add a few drops of glycerine onto it. And now, carefully, using the forceps, transfer the peel into the glass slide and spread it out nicely, evenly using the brush. Next, what you're going to do is place a cover slip on the slide with the help of a needle. You need the help of a needle because you need to make sure that there are no air bubbles. Remove the excess liquid by using a blotting paper on the sides of the cover slip. Ta-da! This sample is now ready to be observed under the microscope. Wow, what do you see? You can see the nucleus as a dark spot in the cell, right?
Yes. So, it was a very nice explanation of that visualization experiment of a cell in which we visualize the onion peel cells. Okay. In the same way, we can also visualize leaf peel or cheek cells or a pond water. In pond water, there are many types of microorganisms. We can also visualize them. Okay. So, yeah, let's also discuss about another experiment. I mean, not experiment, only a simple process part of this leaf peel experiment. Okay. So there are mainly four steps that are needed to be done. First, in the very first step, you need to take a thin layer of epidermis from the lower part of leaf and wash it to remove that dirt, and then keep it in distilled. That glass slide or watch glass. Sir, I also seen animal and plant cell. Okay, okay. So in animal cell, what did you saw? The cheek cells or anything else?
Sir, ma'am has taken the slide and then trying
I
to
mean,
see the
which cells you saw in case of animals? Cheek cells or something else?
sub. I don't know, sir, but in which ma'am stored it in bottle type structure.
Okay, so it it would be cheek cells as it is the only which is in your syllabus at class 9th. Okay, no issues. So first, what you need to do is washing, then keep it wash using distilled water, and then keep it in watch glass or that petri dish. This is called petri dish or watch glass. Then you need to stain it. You need to color it. Why to color it? So as the cells are clearly visible. And while staining, you are using saffronin. While using, what you are doing is you are staining it with saffronin. Saffronin is a dye which is used to color cells. It is very important. Saffronin is a dye that is used to color cells. Then you use glycerine. That glycerine serves two purposes. Okay, add glycerine. Glycerine serves two purposes. First, it prevents the cells from being from drying, and the second thing that it fixes the color. It fixes the stain with that cell. And then there is fourth, that is placing cover slip. Covered with cover slip. And as I said, this is not that clear. Let me write it again. As I said, this cover slip should be used with, placed with the help of needle so as to prevent any air bubbles in it. As the air bubbles may look smaller from your eyes, but when they will be visualized in the microscope, that bubble would be magnified to thousand lakhs of times. So it would be very difficult to then visualize the cell. That's why you need to keep ultimate care. Then when you are visualizing that cells, these all four types of cells, you will be finding two types of cells. In some of the cells, there is a nucleus which is distinctly visible, and you call it as eukaryotes. Yeah, Laksh, you are trying to say something. I can see sometimes you are unmuting and then muting. Yeah,
So nucleus, we said it at prokaryotic cells.
yeah. If the nucleus is absent, you call it as prokaryotic cells. So if nucleus is present, you call it as eukaryote. If nucleus is absent, you call it as prokaryote. Okay. So again, let me call our master tutor so that we can differentiate clearly between this prokaryote and eukaryotic cell.
Prokaryotic cells and eukaryotic cells, and this classification was based on the presence and the absence of membrane bound organelles in cells. So prokaryotic cells are the cells without any membrane bound organelles, while eukaryotic cells have membrane bound organelles. So if prokaryotic organisms do not possess any membrane bound organelles, it also basically means that they lack a nucleus, the most important membrane bound organelle. The nucleus contains the DNA or the hereditary material of the cell. Prokaryotes, however, have an alternate route for this. They have their genetic material, but they have it in direct contact with the cytoplasm. So this is actually the basis on which the term prokaryotic was created. In Greek, pro means before and karyon means kernel or nucleus. So prokaryotic or before nucleus. Eukaryotic cells, on the other hand, possess a well-defined nucleus. And so you have U meaning true and karyon again meaning kernel or nucleus. So eukaryotic or true nucleus.
Yeah, so let's differentiate between these prokaryotes and eukaryotic cells. Prokaryote term is made up of two terms, pro plus karyon. Pro means primitive.
And karyon means nucleus.
Yes. And this karyon means nucleus. In the same way, this eukaryote is also made by these two words, U and karyon. U is,
True.
yeah, U means true. And then karyon means?
Nucleus.
Very good, very good, very good. Now, talking about the major difference in prokaryote and eukaryote. The prokaryote, there is a true nucleus is absent. In eukaryote, there is true nucleus present. Now, what do you mean by this true nucleus? Okay, true nucleus means, yeah, anyone?
Sir, the nuclear membrane is present in any
Yeah,
prokaryotic cell. And
true
in
nucleus.
cell,
Yeah,
the nuclear membrane is not present.
okay,
We
okay,
said
okay.
it at nucleoid.
Yeah, okay, done. And Laksh, your voice is little bit breaking, so you can try changing your room or something like that, or refresh or rewind anything. So true nucleus means actually it has a well-defined nucleus, a nucleus with a nuclear membrane. While prokaryote or primitive nucleus means the nucleus is not that well-defined. There is no nuclear membrane in that organism. And in eukaryotes, there are membrane-bound organelles present. While in prokaryotes, membrane-bound organelles are
Sir,
absent.
so
Yeah,
in
yeah.
prokaryotic cells, the nucleus is exposed to the cytoplasm.
I cannot hear you all, beta. Say it little loudly.
Sir, in prokaryotic cell, nucleus is exposed to the cytoplasm.
Yeah, as there is no nuclear membrane, there is no clear cut boundary between that and genetic material and cytoplasm. So you can say that it is exposed in that cytoplasm. Okay. Now, talking about the cell size of this prokaryotic cell is, yeah,
5 to 10 micrometer.
1 to 10 micrometer, beta. And the
Yes,
cell size of this eukaryotes are 5 to
5 to 100
100 micrometer.
micrometer.
Yeah, yeah, yeah. So, any doubts till now? Anything that you need to ask again?
No, sir.
No, done. So now let's label this prokaryotic cell. Okay, Prachi will be coming to this in later part of this session only. DNA, genes, what are these all? We will be coming to this. Dealing in this session only. So look at the diagram of this prokaryotic cell. This is called
Plasm.
cytoplasm. Then what is this? This is the genetic material. The genetic material is called, in case of prokaryotic organism, nucleoid. Not genetic material. The place where genetic material is there, it is called a nucleoid. Okay. Then you have this cell wall.
And the tail-like structure is called
Yeah.
flagellum.
Wait. There is a doubt. Yeah. Anyone having doubt?
Sir, is this cell wall or cell membrane? Because I know that in plants there are cell walls. Is there?
But
is
it's
cell wall in
not
the
like that. It is only plant which has cell wall. Other than plant, bacteria also have cell wall made up of peptidoglycan. We discussed in the beginning that recap part. Okay. So other than plants, algae, those fungus, those bacteria all have cell wall. It's only animal which is lacking cell wall at your level. Okay. And then there is this flagella. Flagella. Flagella is a structure that helps in movement, locomotion of this bacteria. Now again we are to a poll question. This time I'll be giving you only 15 or 20 seconds looking at your responses. Your time starts now. Which among the following are the examples of eukaryotes and prokaryotes respectively? Let me tell you one thing. Archaea is also a prokaryote. Archaea is other name given to this prokaryote. Yeah, it's Laksh. Others also, give it a try, beta. 15 more seconds. I can see many wrong answers are there. Many wrong answers are there. Others also, 5 more seconds. 3 of you are left, beta. Give it a try. No? Okay, so let me move on to the answer part. The correct answer to this question is fungi and archaea. Now, what is this fungi? Mind it. Other than bacteria, all other organisms are eukaryote. You can write it right over here. Other than bacteria, all others are and archaea is a group of bacteria. Archaea is also a name given to specific group of bacteria. So first one should be eukaryote and the second, yeah, Laksh, ask it.
Sir, blue-green algae is a which type of
Blue-green
example?
algae has got another name called cyanobacteria. And again, the name bacteria is there, so it's also a prokaryote. Got it?
Sir, but in starting we discussed algae is eukaryotic cell.
What did I say? That algae is eukaryotic, that is true. But cyanobacteria is the other name for that blue-green algae. Blue-green algae is only the name. Actually, it's a bacteria. It's only the name. All other algae are, of course, that eukaryotic cells. Got it?
Yes, sir. Thank you,
Okay.
sir.
Yeah. Anyone other having any doubts? Yeah, Prachi, say it.
Sir, what are protists?
Protists are also eukaryotes. Amoeba, paramecium, all these are protists. Okay. So keep it in mind. Other than bacteria, or cyanobacteria is other name for that blue-green algae. So that is not an algae; that is a bacteria only. So all the bacteria are prokaryotes, and all other organisms are eukaryotes. So eukaryote, and one more thing that I added: archaea is the name of a group of bacteria. So fungi is a eukaryote, and archaea is a prokaryote. Okay. Now, again, let me move on to the next question. This time, the question there is assertion and reason. So the first statement: both prokaryote and eukaryotes are well developed. Is it true or false? False.
Sir,
What about the second statement? Eukaryote have nuclear
is
membrane as well as
true.
membrane bound organelle. Yeah, that is true.
One is
So
true.
the correct answer is
True.
3.
A is false, but R is true.
That's it. Now let's move on to the next part. Now, we talked like, okay, so prokaryotes and eukaryotes have so much differences, but there is something in common also. And the thing which is in common is the genetic material. Okay. That nucleus part, the nuclear region. Okay, so again, for explaining for a better understanding of that concept of nucleus, I'm calling our master tutor.
This is how it looks. It just looks like a dark spot, really, when seen in an ordinary microscope. But there is a world of revelation if you were to observe the nucleus under an electron microscope. You would see something as wonderful as this. The nucleus has a double-layered nuclear membrane covering it. There are also some channels that span through the two nuclear membranes. We call those nuclear pores because they look at holes when you look at it from outside and inside. It is through these pores that the RNA and the protein molecules move in both the directions. This is how it looks. It just looks.
Yes, so now let's label the nucleus part. So look over here. Very simple diagram is there for nucleus. So first one is the pore. The pathway through which the movement of molecule occurs inside and outside the nucleus. That is called
Nucleopores.
nuclear pore. What about the second one? This.
Nuclear plasma.
Yeah, it's called nucleoplasm. Okay, it's nucleoplasm.
सर बोलने में हो गया सर सॉरी.
अरे no issues, beta. Nucleoplasm is the cytoplasm of nucleus. Okay, then the core reason, the main reason of that nucleus, it is called
Nucleoid.
nucleolus. It's called nucleolus.
Nucleolus.
Nucleolus. Now, just relate that in prokaryotic organism, that area containing the genetic material was called nucleoid. Here in this eukaryotic nucleus, the core region where the where the ribosomes are synthesized is called nucleolus, and then the protective boundary called nuclear membrane. Yeah, very good, beta. Very fine. Now again we are to this question. We need to state whether the following statements are true or false. So more than one nucleus may be found in a cell. A nucleolus may be found in a cell. Is it true or false?
False.
It's true.
True.
Okay. Actually, there can be more than one nucleus in a cell. Okay. There can be more than one nucleus in a cell, and if there are more than one nucleus in a cell, obviously there can be more than one nucleolus, as each nucleus will contain one nucleolus. Okay, so this is true. There are many
Sir,
cells.
second one is true.
Nucleolus is the site of synthesis of protein. No, beta, this is
So
false.
Sir, ribosome.
Ribosomes are there. That's true. Ribosomes are synthesized. It's true. But the ribosomes which are synthesized are not in turn synthesizing that proteins, and that is why nucleolus is not the site of synthesis of protein. It's the site of synthesis of ribosome. That's true. Okay, beta. So now again we are to this poll question, and let me see who among you is giving me the first correct answer this time. Your time begins now. Okay, one more thing. How many of you cannot visualize that slide? As I can see, many of you are not attempting at all. Poll question.
Last time.
Okay, last time it was visible.
Yes, sir. So we don't get time to solve it.
Okay, maybe it was due to that network issue. Okay, done. So now your time starts now. Which of the following statements are true regarding the function of nucleus? You are left with these 4 statements. Don't be so fast that you get it wrong. Okay, it's Shreya. Shreya who got the first correct answer. Okay, I mean so many wrong answers. It's only Shreya who has got it correct. All others are wrong. Rethink it. Rethink. I'm giving you full time. I mean, left 25 seconds. Rethink. It's a multi-correct question. A multi-correct. 15 more seconds. Now, okay. Oh. 10 more seconds. Okay, Naina also got it correct now. 6, 5, 4, 3, 2, 1. Time up. So the correct answer to this question is A and C. Okay. Just read it out.
I couldn't.
Okay. Yes, Hannah says.
Selecting multiple correct answers.
Yes, sir.
Oh.
Sir, we only click only one option.
Okay.
I
So
choose only A.
okay, Naina, you chose only A. Then it's so correct to me. Okay. Shreya, what did you choose?
First is C.
Sir, even I chose C.
Okay,
Sir,
so
I choose A. Yes, sir, I choose C.
I choose C.
I can see it is only Shreya and Naina who have got it correct. So Shreya chose C. Okay, maybe there is a technical issue, but the correct answer is both A and C. Why is it so? Let me explain it.
Sir, it shows you missed the one. Sir, but I answered the question.
No, I saw that you answered, but you have chosen, the wrong options. Okay, so let me explain. Whatever is the technical issue, let it be. Let me explain. So, nucleus, it controls the metabolic activity of a cell. This is quite easy and true. And then the third one, it contains the genetic information which is passed on to the next generation. This is also true. Okay. Now it's time for us to again call our master tutor. This time we'll be talking about the gene DNA part. Okay. The doubt for Prachi. Yeah.
The nucleus contains the DNA, which we know genetic material that is transferred from parent cells to daughter cells. But what is not widely known really is that DNA is a code that pretty much micromanages all the activities, all the functions, all the interactions of all the components, right from the cell to the entire organism.
Yes, look over here. DNA is actually a double-stranded molecule. Okay, double-stranded. As you can see, there are two strands over here. Okay, and it control. It contains the genetic material, all the codings of life, all the codings. Even you'll be surprised to know that not only coding, even our death is also pre-planned and predetermined in this DNA. Okay, natural death. I'm talking about that natural death part. Okay, so we have programmed cell death. Our death is also our death of death of our cells are also programmed in this DNA only. Okay,
Sir, then we can scientifically know our death date.
beta, what did I say? Natural death. Natural death. Okay, so there may be some other factors for that. Maybe some disease occurred, maybe accident, anything can happen to us, any one of us. The life is totally unpredictable. So the natural death is there in that DNA or that cell, which is coded, but there are many other ways of dying. Isn't it? Okay, now, so it is a double stranded biomolecule. Let me write over here. Double stranded biomolecule. Double stranded biomolecule that contains all the genetic information of a cell. It contains,
Genetic information.
yeah, all the genetic information of a cell. Okay, then the functional portion, there are functional portion of it in our DNA. Keep it in mind that our DNA is a very long molecule. Practically you can imagine, like, okay, the length of DNA is somewhat like half the length of a sari. You can just imagine how long is a sari, and then half of its length is the length of that DNA. Okay, practically, approximately, it's an approximation, approximated length. Then the whole part of this long molecule is not functional. There are only several regions, certain regions that code for proteins, and those regions, those functional segments are called genes. Okay, so genes carry information that determines the trait. Now what is this trait? So traits are the types of characters. Maybe if I say that, okay, I have gene for my, eye color, and my eye color is like black or blue, or often height, that is tall or dwarf. Okay, or skin color, maybe brownish, maybe whitish, maybe black. All these are called traits. Okay, maybe that hair color, black, that brown hair, isn't it? So all these are traits. So genes carry information that determine the trait, the characters that are passed over generations, that are passed over generations. Now let me also give you a certain idea of that organization of DNA into a chromatin and chromosome. Now let me tell you that DNA is nothing different from those chromatin and chromosome. All of them are same thing. Okay, the thread is analogous to that DNA. When you just mingle that thread, just entangle it, then it will form chromatin. And when you organize it, that thread is so long, and when you roll it over on the thread roll, it's so organized. It is the same way that chromosomes are formed. Chromosomes are nothing but DNA, a well organized DNA. Okay, so let me first tell you what is this chromatin. So chromatin is the uh, entangled mass of thread like structures. Entangled mass of Let me write over here structure. Which up? Oh, sorry. Structure is there written, already written. Okay. So entangle mass of thread-like structure present in the nucleus. Okay. Yeah, Prachi.
Chromosome.
Yeah. Then talking about this chromosome, as I said, that entangle mass of thread, if it is again rolled in inorganized way, it forms chromosome. So chromosome is nothing but chromatin, which is organized, which has organized itself to form chromosome. Okay, nothing but chromatin organized to form a chromosome. Now, let me tell you one more difference, that prokaryotes have only one chromosome, while the eukaryotes have more than one chromosome. This is also a major difference between that's prokaryote and eukaryote. Now, talking about that chromosome. Okay, so chromosome is only visible when the cell is going to divide. Otherwise, you cannot see chromosome. Normally, the DNA is there in the form of chromatin. Okay, that chromatin material. Uh, and when the cell is going to divide, only then the chromatin organizes itself to form DNA. So, chromosomes are something like condensed genetic material. Okay, there is one doubt. Okay, yeah. Condensed genetic material that are coiled into rod-like structures. Coiled into rod-like structure. Look over here, the rod-like structure. Okay, so chromosomes are only visible when our cell is going to divide. Otherwise, it is not visible. Normally, the chromatin fibers are visible. Okay, so now, Prachi, is your doubt clear about that DNA, gene, and cell? Prachi?
Yes, sir, it's clear.
Okay, done. So now let's solve this question. This is the same question that you were asking, but in some different way. They're asking that you need to, arrange all this in ascending orders of the organization. Okay. So the smallest one is this gene. As you know that even in the DNA, the functional part is called gene.
Sir, first gene, then chromosomes, then
No, then DNA.
okay,
Gene,
sir,
then
then
DNA.
DNA, and
Yeah,
then
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 chromosomes, and then
chromosomes, then
nucleus,
nucleus
and
and cell.
then cell.
yeah. Now
Last
it's
is
fine.
cell.
Now it's fine. Yeah, very good. Now let me just again differentiate between this prokaryote and eukaryote as a summary part. So size, prokaryotes are smaller, eukaryotes are larger. Nuclear region, not well defined. Okay.
Nucleoid.
Ha. And then eukaryotes have well defined nucleus with nuclear membrane. Chromosome single in prokaryote, more than one in eukaryote, and membrane bound cell organelle absent in prokaryote while
Present
Eukaryotic
in
present.
eukaryote.
present in eukaryotes. Very good. So now, but aware to this doubt board. So anything that you need to ask? Anything?
No, sir.
Sir,
Yeah, Prachi.
Sir, what does RNA stands for?
RNA. Okay. RNA is called ribo. Okay, Sahana, I'm coming to you. Nucleic acid. In the same way, what is this DNA, Prachi?
Sir, it's deoxy,
Yes.
deoxy, nucleic acid.
Deoxyribonucleic acid. Deoxyribonucleic acid. Okay.
Okay, sir.
Let me write over here. Deoxy.
Difference between them.
Yeah. Nucleic, CLEIC, nucleic acid. Yeah, difference is that RNA is less stable, DNA is more stable, and there are many other differences as well. DNA is double stranded, RNA is single stranded, and many others are also there, but you will be discussing it in higher classes. Okay.
Yes, sir.
Yeah, done. Yeah, Sahana, now ask your doubt.
so why do we use glycerin while examining these cells?
Glycerin serves two purposes. First, it fixes the color, that dye. It fixes the color so that when a cell is being colored at one time, it will not lose its stain after some time. And
Okay,
the
sir.
second function is that it will, not allow that cell to dry.
Okay,
Okay,
sir.
done. So now let me now discuss prepare an analogy, and we'll be discussing about preparing an analogy like cell is analogous to a chocolate factory. Okay, I mean, in that cell, there are many types of chocolates which are being formed. Okay, so you know that in a cell there are many things which are taking place simultaneously. So in that way, we'll be calling it, okay, for preparing a chocolate, what we all need in a factory. First, there will be a recipe. I mean, all the rules and regulations that what you need to do after each step. Then there must be a chef that would be preparing that chocolate. Then there would be a production a layer, I mean, a production flow in which the specific chocolate is being formed in that factory. And then there is one man who will be processing, packing it, and so that it can be sent to the market and sold. In the same way, there are four other things in the cell also. First, there is something called recipe book, and it is our DNA, the genetic material in our cell. As I said, that genetic material contains all the information of every protein that is being made in our body. It has information for all of them. Then, so you can simply say that, okay, it is this genetic material is analogous to that recipe book in chocolate for chocolate products. Okay, now we have that recipe, but we need that sieve so as to produce that chocolate. In the same way, we have that genetic information, we have that coding, but there is a need of something or some organelle that can decode that coding and prepare that protein using that code, using that information, and that function is done by this ribosome. Okay, so now uh, let's look over this ribosome. Ribosomes are membrane free organelle, that are membraneless organelle. Those are associated with protein synthesis. Now let me tell you one more thing, like, okay, if I say that membraneless organism, so there are only two organelles in a cell that are membraneless. One is ribosome and the other one is centriole. Centriole helps in cell division. We'll be dealing about the centriole in later sessions. So centriole membraneless, and then ribosome, which is
What?
membraneless.
Centriole is present in only animal
Animal
cells.
cells, yes, it is present only in animal cells. We'll be dealing about that centriole in higher classes. I mean, not higher classes, maybe in our later sessions also. So ribosome and centriole are the only membraneless organelles, and this ribosome is responsible for protein synthesis. Now we are to this question. This time the question is, which of these are not the function of ribosome? Okay, so the first one, it helps in manufacture of protein molecule. This is true. Second one is it helps in manufacture of enzyme. This is also true, but keep it in mind that most of the enzymes that we have in our body is protein. Most of the enzyme that we have in our body is protein. So the proteins are, as they are enzymes, so they are also made in
Option C
this.
is incorrect.
Yeah. So options, what? Let's come to this option C. It helps in manufacture of hormones. Wrong. Hormones are not enzymes; they are lipid or fats or steroids. We'll be learning about that in later classes. So these are not proteins only. So this is wrong. It helps in manufacture of a starch molecule. This is also false. So the correct answer is both C and D, as they're asking for the wrong statement. Okay. Now let me provide an analogy to this endoplasmic reticulum. As I said that, okay, first you have that genetic information, then you have that ceph, and now you need a floor, a place where this production is taking place. So in cell we have this endoplasmic reticulum on which there is ribosome, and that ribosome helps in protein synthesis. Okay, so now
But sir, in the smooth, in the plasmic reticulum, there is no. Wait, sir.
so smooth endoplasmic reticulum is not associated. We'll be discussing it. I'm actually calling our master tutor who will be discussing it about all these SER, RER, and which endoplasmic reticulum is actually responsible for that ribosome, which helps in protein synthesis in turn.
So what
So
is
yeah,
Golgi bodies?
we'll be coming to it in later part. I mean, just after 2-3 slides. Okay. Yeah. So it's time us to call for our master tutor.
There are different kinds of structural elements. You've got cisternae, which are long, flattened, sac-like structures. These structures usually lie parallel to each other. Then you've got vesicles. Vesicles are nothing but round or oval membrane-bound sacs. They're usually present individually away from the rest of the network. There's also the tubules, which you can guess from the name, are tube-like structures present in the endoplasmic reticulum. They're usually present as branch structures, if you think about it, similar to a blood vessel, right? Now, there are two types of endoplasmic reticulum, a rough one and a smooth one. There is a region of the ER that is continuous with the nuclear membrane, and it has a lot of ribosomes on it. That's called the rough ER, since it's not smooth, but it's irregular and granulated because of the ribosomes. You also have regions of the endoplasmic reticulum where there is no ribosome lining, and that is what we call the smooth ER. Now, while the rough ER is continuous with the nuclear membrane on one side, it usually, as you can see, transitions into the smooth ER as it moves away from the nucleus. So the other main difference between the two types of ER is that the rough ER is going to be more actively involved in the synthesis of proteins and enzymes, which is pretty obvious because, as I told you before, the presence of the ribosomes is pivotal to the synthesis of proteins. So the rough ER is usually more abundant in cells that are actively involved in making more of protein. And on the other hand, the smooth ER is more involved in the synthesis and storage of lipids like steroids, cholesterols, and steroid-based hormones, and also sometimes in the synthesis of glycogen. Do you know what's glycogen? Glycogen is the form in which glucose is stored in humans and animals. There are different kinds of structural elements.
Yes, so this is the endoplasmic reticulum, as I said, that we all discussed. So there are two types: smooth ER, which manufactures lipid. Okay. So lipid, and then there is rough endoplasmic reticulum, which manufactures
Protein.
Protein.
yeah.
And enzymes.
Any doubts, beta?
Sir, I was telling proteins and enzymes.
Yeah. That enzyme or that lipid. Then there is something like this. There are three parts: tubules, Christie, and vesicles. Tubule, tube-like structures similar to blood vessels. Christie, long, flattened sac-like structures, and then vesicle, round or oval membrane sacs. Okay, so now do you know that smooth endoplasmic reticulum also helps in detoxification? Detoxification means the toxins which are formed after eating, I mean, something wrong, like maybe alcoholism, or sometimes like any drug or toxin which knowingly or unknowingly entered your body. So it converts all that drug and metabolic wastes into water and other compounds which are less toxic or no toxic, and hence allows safe excretion. Then there is one NCRT question. The question is, where do lipids and proteins synthesized in our lipid and protein constituting the cell membrane get synthesized? Okay, so lipid is synthesized in SCR while protein is synthesized in RER. Now let's talk about this Golgi body. As I said that after all that production is done, you need a person who is actually packing it and transporting it. So it's that Golgi body. And again, to know more about this Golgi body in detail, I'm calling our Master Yuta.
The Golgi complex is the cell organization of packaging and tagging of the biomolecules that are obtained from the endoplasmic reticulum for further transport. So the Golgi complex also has many names like Golgi apparatus, Golgi bodies, and even dictyosome. The Golgi complex was named after the scientist who first discovered it or found it, Camillo Golgi. In fact, it was one of the first organelles to be discovered because it's such a huge and prominent part of the cell, you couldn't miss it. And as you can see, the Golgi complex is made up of a bunch of flat, expanded, membrane-bound discs stacked together. We call them cisternae. Now, these structures are very similar to the structures that you found in the endoplasmic reticulum, which is why we just give it the same name for simplicity, cisternae. If you were to imagine that you were in the nucleus at the center of the cell, Surrounding the nucleus, you have the endoplasmic reticulum. And after that, you'll have the Golgi complex. There would be some cisternae of the Golgi complex facing you or the nucleus. These cisternae would be convex and is called the forming side or the cis cisternae. This is the side that receives secretory materials through the transitional vesicles from the endoplasmic reticulum, as you can see over here. What next? The materials are then packaged and labeled as they travel through the layers of the Golgi. So what does the packaging and labeling mean over here? Of course, the proteins are not going to be wrapped up in colorful paper or brown paper with addresses written on them. They are going to be labeled with specific biomolecules. For example, proteins that are tagged with a derivative of a carbohydrate called mannose-6-phosphate are destined to the lysosomes. Proteins that are sorted and tagged are going to leave the Golgi body in vesicles from the concave side, which is called the maturing phase of the transcisternae, and they continue their journey from there. They are then taken to their specific target organelles inside the cell. Some others head to the cell membrane, and then some more for secretion. The vesicles that are released through the transcisternae are transported towards the membrane, where they may either be integrated into the membrane or secreted outside the cell. The Golgi complex is the cell organelle.
Yes, so let's discuss the function of Golgi body. First, there are two types, as we just learned. Some are called cisternae, while the others are vesicles. So, talking about that cisternae. So, cisternae are flattened disc-like structures. Let me write over here. Flattened disc-like. Okay, flattened disc-like structures where the proteins are labeled. Proteins are labeled and it is modified. Then there is vesicles. So in these vesicles, there is transportation, or you can say synthesis or packaging. Synthesis and packaging. K-A-G-I-N-G. Synthesis and packaging to various parts of the cell occurs. Okay. So now again we are to this question. This time the question is, which of the following features are true, functions are true for Golgi complex? So the correct answer is storage.
Packaging of materials.
Storage is not there. Storage is not there. Storage is not there. Keep it in mind. Storage is not there. Packing is true, but storage, yeah, it's true. And C is also true. C is also true. Synthesis of lysosomes also occurs. Lipids are not synthesized in Golgi bodies. They are synthesized in SCR. Now we are to this textbook question. This time the question is, what would happen to the life of a cell if there is no Golgi apparatus? So beta, if there is no Golgi apparatus, there cannot be formation or packaging or modification or transfer of proteins. Okay. So sometimes these proteins or lipids are very important for plasma membrane synthesis. So it will be hampered. And if there is no more plasma membrane, there is no new cell formation, as there is no cell division. Again, one more question is there. We need to match the following. Nucleolus. Nucleolus is control center of nucleus. Endoplasmic reticulum.
Synthesis of proteins.
Synthesis of lipid. Better lipid. Ribosome is responsible for synthesis of protein. And then Golgi complex. Packing of
Packaging.
very good, very good, very good, beta. So any doubts till now? Anything?
No, sir.
Okay, done. So let me now revise all what we learned today. We talked about that prokaryote and eukaryote cell. We talked that prokaryote has primitive nucleus, eukaryote has modern or well-developed nucleus with nuclear membrane. membrane bound organelles are absent in prokaryote while present in eukaryote, and the prokaryotes are smaller while eukaryotes are larger. Now talking about that gene DNA. So DNA is the basic material, the double stranded structure that has all the codings. The functional part of the DNA is gene. The DNA entangles to form. Entangled thread-like structure are formed by this DNA to form chromatin fibers. And when these chromatin fibers are organized in rod-like structures, they are called chromosomes. Okay, and chromosomes are only seen in the nucleus at the time of cell division. Then we talked about endoplasmic reticulum, two types, RER, SER. RER lipid synthesis, RER protein synthesis, and SER in lipid synthesis. Nucleus, the control center for the cell, and then it has nucleolus, the core region of that nucleus, which synthesizes ribosome. Then we talked about ribosome present on RER and in cytoplasm also, and helps in protein synthesis. And then there is Golgi body that helps in packaging and transport of protein inside or outside the cell. Okay, beta. So this is all for today. This is me, Atul Kumar, waving all bye bye. And yeah, please come back and practice more and more questions. So
Bye,
anything,
sir. Thank you, sir.
yeah, anything else that you need to ask, or should I end up the session?
No,
Anything?
sir.
Okay,
No,
done.
sir.
Bye,
Done.
sir.
So bye bye. So this is all for today. This is me, Atul Kumar, waving all bye bye.